Hello, Dr. Eugene Aiello here. And thank you for watching my next video series on patient education. This is going to be an exciting video. We are going to talk about how cells function and how biophotonics relates to cells and what biophotonics is. I hope by the end of this video that you understand more about how a cell functions and how life exists than the average doctor that you would see. This area is going to touch on breaking some of the conventional models of cellular physiology. I talked last video about the Rockefeller industry and how everything is deterministic steps of biochemistry. And as I described some of how a cell functions, I think you're going to understand that it doesn't make a lot of sense. There's something driving those chemical reactions. That is never answered in conventional biology. As a biology student, we learned quite a bit about these things, and you just would sit there and think, how does this happen? It's not just random events that these physiologic reactions are occurring. Something is controlling this. Something is driving these reactions. So once I'm done with this, I'm hoping that you'll understand a lot more about the flaws of the conventional system and how biophotonics is the answer to most of those questions. It explains how these different cellular activities occur. So I'm going to first go over the basic structures of a cell. This, again, very basic. You've got your nucleus in the middle here. Your nucleus is holding your DNA. Your DNA is purely just templates for proteins. I think the average person somehow thinks there's some type of memory that the DNA controls the cell and that's the center of the cell, when all it really is holding those amino acid sequence templates that are later used to make protein structures. You've got your cell membrane. On the inside of your cell membrane, you have something called a peripheral cytoskeleton. It's protein structures that hold the shape of the cell, but more importantly, deals a lot with the different ion fluxes, how things go across that membrane. In that cell membrane, we also have transmembrane proteins. There's multiple types. I didn't put these in there, but conventional science talks a lot about pumps and channels. Those pumps and channels control that ion flux through utilization of ATP energy sources. We're going to briefly touch upon that this is a completely flawed model. It would take too much energy to consume to maintain the different ion gradients. Those ion gradients are going to be controlled by the semiconductor nature of proteins, which I'm going to talk about in a little bit. So transmembrane proteins, cell receptors, different channels that allow things to come and go out of the cell. You've got mitochondria in the cell. Mitochondria are the centerpiece of my model. They're what produce ATP. ATP, adenosine triphosphate, is the stored charge potential that cells use to do everything. Activities of enzymes, flawed models in convention that talk about pumps and channels require massive amounts of ATP. So ATP is how different things get accomplished. And that's just one way that they get accomplished. But in conventional models, that's the only way. So many of the things that happen within a cell would require vast amounts of ATP, way more than the cell would need when you take into account what biophotonics are accomplishing. Next to the nucleus, with protein production, so when those protein templates are made, the DNA will open up to certain segments that are nucleotide sequences that match up to different amino acid sequences. Each cell is going to open up that DNA strand at different places to exhibit the proteins necessary for the function of that cell. That's another unanswered question in current models that I think biophotonics is going to answer. So those DNA sequences will split open and will make mRNA templates. The mRNA is just copying that DNA segment. That mRNA template is then going to be used to make amino acid sequences, just basic chains of amino acids. The next structure is your endoplasmic reticulum. Your endoplasmic reticulum takes those simple amino acid chains and modifies them and turns them into more complex proteins, which eventually become enzymes, different structures within the cell, transmembrane proteins. All the different things that the cell has are produced from those templates, and then they're assembled in the endoplasmic reticulum. The endoplasmic reticulum will take those final proteins and ship them to the Golgi organ. The Golgi organ is responsible for packaging and labeling that cell of where it's going, or that new protein, where it's going to go in the cell. Which brings us to our next system, which is the microtubule system. In conventional science, the microtubule system really has some pretty minimal functions to it. In the biophotonic model, it's the centerpiece. That microtubule system is going to be the absorber of biophotonic energy, and it's going to be the processor. Within these tubule systems is all the data that's in that cell, and different frequencies of energy are going to express that data differently. That different expression of data is going to relate to genetic expression. It's going to relate to how this cell maintains itself. So that microtubule system, again, will spread throughout this entire cell and will be responsible for the structure of the cell. Cells, simple cells like bacteria, will use those microtubule systems to make finger-like projections and move, the pseudopods. So at the center of this microtubule system is something called the centrosome. The centrosome is composed of two parts, a 90-degree orientation of two proteins. These proteins are microtubules that are very complex. They're multiple microtubules in sets of 3 triplets. 9 of them construct the one centriole. The 2 centrioles are completely different. The mother centriole is extremely complicated in its structure. That is the only structure that's going to link to these microtubules. So it has appendages that have microtubule branches off to them. The daughter centriole will only come into play during mitosis. During mitosis cell division, these two will split off, and that daughter will mature into a mother centriole, and then it will divide the cell. So in the center here of the centrosome is interconnecting fibers of proteins. So there's a lattice network of proteins between these 2 centrioles. Coming from that lattice network, we have microtubules that project throughout the cell. We even have microtubules that project right into the DNA, right into the nucleus of the cell. So those microtubules there are responsible for the unfolding of the DNA and the splitting of DNA during mitosis. So we copy our DNA, spread it. Okay. Another thing I want to point out is these microtubules are associated with that peripheral cytoskeleton. They're also associated with these transmembrane proteins. So they can gather information or send information possibly through the system, which I'll detail a little more later. Oh, and I forgot to mention this Golgi tendon organ, this Golgi organ. When it's done packaging that different new protein that was produced, it creates something called an exosome. Those exosomes are the packages that have the instruction for where that protein is going to go in the cell. Some of those proteins are excreted. They could be hormones, they could be communication, they could be as simple as tears from a cell that's producing, some type of excretion. So these exosomes are carried by those microtubules, and that's what decides where those different proteins that are just produced from the DNA are located. So that's the basics of a cell. So what is a cell's function? A cell is going to produce proteins, protein synthesis. I kind of touched on it already. DNA is the template. It's going to create the proteins. What are these proteins for? Well, I discussed the cytoskeleton, structure of the cell, movement of cells. So muscle cells will have contractile fibers produced by proteins. Enzymes. Proteins are the catalysts. What an enzyme is an enzyme will take 2 different products, join them together, and create a new process. Catalysts make a reaction occur rapidly. In theory, 2 products that could interact together may do that over time, but it may take hours, years, whereas a catalyst has that reaction happen rapidly. So metabolism. So when we're talking about sugar metabolism, we break things down for energy. Enzymes are responsible. When we're constructing things, making amino acid segments, there's going to be enzymatic activity that occurs within this whole process. Enzymatic activity to produce fats, to take different simple fats and combine them for making the cell membrane. So enzymes build and they break things down within the cell. Proteins can be used for communication. Chemical. When I say chemical, I'm talking conventional models. We're going to discuss that chemical nature is a flawed theory within that system. What I mean by that, when we created this exosome that we excreted, it possibly could be a hormone. Hormones are thought to interact with these cell receptors on other cells. So a hormone is secreted by, say, the pancreas is secreting insulin. That insulin is going to flow through the blood. Conventional theory says that the interaction at the cellular level is just purely lock and key fitting of that insulin molecule to a receptor. That is an extremely flawed model. It's what's taught in this Rockefeller system because everything's interactions of chemicals, remember? But insulin floating through the blood, there would have to be such a large concentration to serve every cell. That insulin would have to leave the blood and get into the lymphatic system, which is a very slow moving fluid system, for it to interact. In the biophotonic model I'm going to propose, that hormone is doing nothing more but sending off a certain electromagnetic frequency. That electromagnetic frequency is received by these receptors. These are acting as antennas to a specific frequency. So an insulin receptor would be tuned to accept that frequency insulin's emitting. That frequency, again, would go into this microtubule system for processing. So that microtubule system is the centerpiece of all cellular function. Communication chemical and receptor, we just discussed, act as a semiconductor. So proteins, by nature, the way that they function is their semiconductor nature. When I said that enzymes are doing things, and I said we learn about all these biochemical reactions which are driven by enzymatic activity. Enzymes are doing this just how? So what I'm getting at is it purely just that they fit the different substrate A and B and they bring them close together and they make the next compound? That's not at all what's happening. When those compounds are made into the next structure, there's chemical interactions between electrons. There's joining of molecules based on dynamics at the quantum level. So semiconductors, proteins can transmit electrons in a nature that's different than a wire. So in a wire, when you have a voltage across it, electrons are going to travel that voltage gradient, but they don't all travel the same direction. Electrons can bounce up and down, even some can go opposite the gradient. With a semiconductor, electrons can only flow in one direction. So if you put a voltage across a semiconductor, those electrons only have the option of going one direction. The reason for that is those electrons are getting excited and jumping to the next molecule. As they jump, something very interesting happens. When those electrons settle in, they will create electromagnetic energy. Did you catch that? So as those proteins have electrons flowing through them and they're moving through, they're emitting biophotonics. They're emitting light. So proteins within cells have the ability to create electromagnetic energy that they produced from a charge separation. So a voltage separation across a protein, as the electrons flowing through it, it can emit biophotonic frequencies. Within this microtubule system, quantum biologists have found that this microtubule system is responsible for light emissions. Every cell is emitting small amounts of biophotonic energy. Conventional science believes that biophotonic energy is just a side effect of all these chemical reactions that are occurring within a cell. But quantum biologists are starting to find that it seems like it's an orchestrated event and it's coming from that microtubule system. So the semiconductor nature of this microtubule system can emit electromagnetic frequencies, and they can emit it specifically depending on the structure of that microtubule. Semiconductor nature of a protein also allows it to absorb electromagnetic energy. So in our pigment systems of our skin, in our mitochondria, there's pigments that when electromagnetic energy absorbs into those, it creates a charge separation. So a solar panel, that's exactly what it is. A solar panel is a semiconductor that takes in sunlight and creates a charge separation. If you hook that to a battery, now you got charge and some potential. If you hooked it up to another semiconductor, which is what an LED light is, if you hook it up to another semiconductor, as that electron flows through that other semiconductor, it's bouncing electrons and emitting frequencies of light. Proteins exhibit those same natures. They're more complex than what we do in our electronic devices. Mother Nature's design is quite amazing. So there is an interaction of electromagnetic energy within proteins. There's also electron flow through proteins, which can change how those proteins are shaped. It can change the dynamic of a protein. So when I'm talking about the different ion fluxes, those ion fluxes are occurring because a protein configuration changes from charged or not charged, when they're potential or not potentialed. That's going to create ion fluxes, which I'll discuss that more when we talk about structuring of water. So proteins have some quite amazing little activities they perform in the day-to-day life of a cell. They control everything that's occurring, and electromagnetic energy and voltage is what's driving how they function. So single cell. If this was a single-celled organism, it would get its electromagnetic energy directly, solar. So this cell can get every different frequency it needs and absorb it into its pigment systems, in its mitochondria, in different amino acid sequences that absorb different frequencies of light. What about a human? Multicellular organisms, we have mitochondria deep within our body. That mitochondria, as we're going to talk about in a minute, is dependent on electromagnetic energy for charge separation to drive that production of ATP. I believe there's an indirect system. I think our pigment system is absorbing those frequencies and putting them into a system within our body that parallels our nervous system. I think I talked about that in the first video. The glial system is transmitting these frequencies into a central system, which is distributing these different frequencies. At the center of every multicellular organism is that centriole. Single cells do not have a centriole. Single cells have no need to receive any frequencies from a system, an indirect system. They directly absorb it right into their system. So multicellular organisms literally have an antenna within the center of their cell. That antenna is going to be specifically tuned to absorb a certain combination of frequencies that elicit the function within that microtubule system. So I'll say that again. When they're absorbing, this cell is absorbing frequencies from this indirect system, which can have multiple frequencies within it. Every cell is going to receive those energies, those frequencies differently. That different frequency absorbing into this microtubule system is going to exert different data within the system. So when we talk about how does the DNA know to unravel that segment to produce certain proteins? It knows that because of the different electromagnetic frequencies that microtubule system is producing. It doesn't carry information. The information is within the proteins, but proteins react differently to different frequencies of electromagnetic energy. So quite an amazing concept. My centerpiece of my research early on was on the mitochondria. Research on the mitochondria shows that when you shine red light on a single cell, it's going to increase ATP output. If you shine radio frequencies, electromagnetic pollution, cell phones, different EMF fields, the mitochondria is going to exhibit the exact opposite. It's going to produce less ATP. So I started to do my research trying to figure out how does electromagnetic energy have anything to do with mitochondrial function. According to conventional models, which I'm going to discuss in detail in the next video, oxidative stress, according to mitochondrial models, electromagnetic energy has nothing to do with it. The mitochondria is functioning just by moving electrons through different proteins, creating a gradient of charge which drives ATP production. Multiple flaws in that model. They never discovered the actual enzyme that creates ATP from ADP. They never proved that that gradient that's being produced ever exists. And the only reason we're consuming oxygen in that model is to accept those electrons that are bouncing through the proteins at the end to create water. So yeah, next video I'll get more into that if I lost you. So within the mitochondria, well, first we'll start off with glucose from the beginning. Anywhere within this cell cytoplasm, we can take glucose and break it down into 2 pyruvates. Glucose is a 6-carbon ring. 2 pyruvates are 3-carbon structures. So in this enzymatic process of breaking that pyruvate down, that glucose down to pyruvate, we're going to create 2 ATP only in that process. That's barely anything. That glucose is going to make 36 ATP by the end of this whole process. So enzymatically, we only create 2 ATP, but we create 4 NADHs, I believe. NADH is an electron carrier. It's holding an electron. So in this enzymatic process of breaking glucose down, they're pulling an electron and they're storing it in that NADH molecule. That NADH molecule is going to become very important in the mitochondria in a minute. That pyruvate then has to enter the mitochondria, and it goes through a cycle called the Krebs cycle. That Krebs cycle is just breaking those carbon bonds down, producing carbon dioxide. So that 3-carbon sugar is going to produce 3, for each pyruvate, 3 carbon dioxides. So a total of 6 carbon dioxides get produced in the process. If you notice, there's no oxygen used at this point in the reaction. So all of this is enzymatic. So this enzymatic has taken sugar and broken it down to carbon dioxide, only created 2 ATP, but a bunch of NADH, which is going to be used in the next step in the process, which does require oxygen. I have a little note in here too about fatty acids and amino acids. They can also enter the Krebs cycle just like pyruvate, and they're broken down into carbon dioxide and producing those NADHs for later. Those NADHs we'll move to another part of the mitochondria called the electron transport chain. So in this inner membrane, there's proteins in a series of 4. This is an example of one of those proteins. Within that protein is a molecule called a cytochrome. Cytochromes have at the center of them a heme molecule, just like hemoglobin, which binds oxygen. The other interesting thing about a cytochrome, it absorbs light. So every different study on red light therapy, trying to understand why does red light increase ATP production? The reason that they came up with, which conventional science, is that that red light vibrates that molecule. It's at the right resonant frequency to make that cytochrome do what it's supposed to do. And in their model, that cytochrome is only accepting those electrons from NADH. So I'm sorry, I should put this in here. NADH will give an electron to that cytochrome and become NAD+. So that electron in the conventional model just bounces between those different cytochromes in that chain of 4. And as it's bouncing, it's creating a charge separation between that inner membrane and the matrix of the mitochondria. The final step of this process is those protons discharge and they produce ATP. Magically, somehow. They discharge through a protein that is supposedly a wheel. And as those protons discharge through that protein and they make that wheel spin, that mechanical energy is converted to chemical energy. Okay. So they believe they found the enzyme 3 times, they didn't find it. So that conventional model is not explaining how this red light is enhancing that ATP function in my mind, because their model doesn't account for reactive oxygen species. And what I mean by that is when you shine red light on a mitochondria, it's going to increase your ATP production, but it's also going to create your reactive oxygen species. Reactive oxygen species in the conventional model are a byproduct, an inefficiency. Reactive oxygen species occur when those electrons are bumping through those cytochromes if they escape too soon. So if those electrons don't make it through the whole chain, they're going to create reactive oxygen species. The final flaw of the conventional model is that that last protein, that last cytochrome that those electrons are bouncing through, the only reason that we're consuming oxygen in that model is oxygen accepts that electron controlled and creates water. Well, if you think about it, that doesn't add up very well. For every oxygen molecule that we inhale, we're creating a molecule of water. That's a lot of water. So between the not having the enzyme, not having that gradient they talk about, and all this water created, that conventional model doesn't make sense. And it really didn't make sense when you're finding that red light therapy is enhancing the function, but increasing ROS. So basically they're saying red light increases the production of it, the efficiency of the mitochondria, but at the same time it's creating a lot of byproducts, inefficient byproducts. Radio frequencies have the opposite effect on ATP. They're decreasing ATP, but magically they're also increasing ROS. ROS, reactive oxygen species, are what cause oxidative stress. Oxidative stress is damage to the cell. The basic aging process of a cell is dictated by ROS. That oxygen molecule that's holding onto that extra electron, it's like a hot potato, meaning it doesn't want to hold that electron very tightly, not like ATP does. ATP stores that electron charge in a very controlled manner. So reactive oxygen species will donate that. They'll donate it to microtubules. They'll donate it to the endoplasmic reticulum. They'll donate it to DNA. When they do that, they denature it. They change the structure of the molecule, which damages it. That's why it's called oxidative stress. So the negative effects of radio frequencies on cellular activity is oxidative stress. So I'm kind of at an odds of how you get reactive oxygen species elevating in both cases. It makes no sense. Tells me that there's a major flaw to the conventional model of mitochondrial function, especially in the electron transport chain. I was doing a little research and there's an alternative model that's coming out called the Merburn concept. The Merburn concept says, no, none of that is occurring. The water, the lack of the enzyme converting ATP, and the lack of any proof that there's even a gradient produced within that membrane. Merburn model says this NADH is donating that electron to that cytochrome, which is binding that oxygen. that cytochrome is donating that electron to oxygen on purpose. It's creating reactive oxygen species because reactive oxygen species are the fuel. Reactive oxygen species are the ones that interact with ADP bound to these proteins, creating the ATP. So reactive oxygen species have to be produced by these cytochromes at a certain concentration to make this equilibrium of the reaction occur. So if everything is going well and those reactive oxygen species are produced at the concentration necessary for ADP to ATP conversion, that reactive oxygen species is getting neutralized. If this concentration reactive oxygen species production is off, you're going to start having reactive oxygen species escaping, which we call diffuse reactive oxygen species. Those are the dangerous ones. Those are the ones that are going to cause harm to the cell. Within our mitochondria, the mitochondria produces an antioxidant system. We consume antioxidants in our food. We understand that it's healthy. It prevents cancer, prevents aging, all these different things. Antioxidants will neutralize those reactive oxygen species. The bad news is our dietary consumption only accounts for about 3% of our total antioxidant capacity. The mitochondria produces a lot of antioxidants. So these dross are buffered to the extent that the system can do it. If the system is overwhelmed by reactive oxygen species, we start to have oxidative stress. So a lot to swallow there. Hopefully you followed all that. So NADH is an electron donor to the cytochrome, which creates the ROS. How does red light play in this? That's the interesting thing. These cytochromes are pigments. They absorb that electromagnetic energy. When a molecule absorbs electromagnetic energy, I mentioned before with the semiconductor nature, if they absorb electromagnetic energy, what they're actually doing is creating a charge separation. How does that occur? In molecules, electrons float around the nucleus. Those electrons can sit at a very outer part of that nucleus, which is the less, how should I say, controlled. The electrons closer in nucleus are tightly held. That valence electron shell, that outer shell, is what's absorbing that electromagnetic energy. When an electron absorbs electromagnetic energy, it excites it. It makes it more likely that it's going to leave that nucleus and get away. That's the difference between ionizing and non-ionizing radiation. Ionizing radiation, UVB light and above, has enough energy in the electromagnetic energy to bump electrons free. When it's bumping electrons free, it's doing the same thing that we have here. You get free floating electrons and you're interacting with proteins, interacting with fats, interacting with DNA, it's going to cause problems. So that's why UVB light and above is carcinogenic. Non-ionizing radiation does not have enough energy to it to bump an electron, but it has enough energy to excite it. So that excited electron is going to have that likelihood to interact with oxygen to create oxygen species, reactive oxygen species. So this red light is coming in because it's the energy that creates the charge separation that allows that donation of oxygen to make the reactive oxygen species, which creates the reaction to make ATP. Mouthful, huh? Well, huge. ATP production is what keeps the cell alive. ATP is a stored way of using that charge separation within proteins for different functions within the cell. So how does this happen? Where we get that inversion, that ATP production goes down, but ROS goes up. ATP production goes up as ROS goes up. What's occurring there is the measurement of ROS. Where are they measuring it? Around this cytochrome, we have this area that in the Berberine concept they call a MER zone, almost like a cloud of these reactive oxygen species. That cloud has to be that perfect concentration for that ADP-ATP reaction to occur and consume the reactive oxygen species. The buffer system's there, those antioxidants are there to deal with it at a certain level. Depending on the frequency of red light, depending on the energy within that light, depends on how quickly this oxygen is going to be produced, reactive oxygen species. So when radio frequencies interact with the mitochondria, it's going to set this out of tune. Those reactive oxygen species might get produced at too slow of a rate. They might be produced at too high of a rate. If they're not at that sweet spot of concentration, you're going to get diffusion of that ROS, and you're going to start to have problems. That's what oxidative stress is, and that's the topic of the next lecture. So Biophotonics. Biophotonics is the study of light emitted in cells. Like I said a minute ago, it's only believed to be a side effect reaction. After you understand how the mitochondria is dependent upon this system, then we start to go, there's something more to this. If this one interaction is so vital at producing ATP, how many different interactions with genetics, how many interactions with enzymes? These are all absorbing these electromagnetic frequencies at a specific range that they function at. And if those frequencies are emitted by the microtubule system, it controls all cellular activity. So that indirect electromagnetic energy absorbing into that centriole kicks this whole system into gear and gets this whole cell functioning. So quite exciting to me. Hopefully you could tell in my tone on this lecture versus the others that this is an amazing discovery. It's basically explaining how all the different protein interactions occur. They're occurring based on absorption and emission of electromagnetic energy. All that energy, by the way, is within the near-infrared, visible light spectrum, and part of the UV spectrum. So get back to our cell function. We talked about protein synthesis. We talked about energy production. We didn't talk about homeostasis, but indirectly I already discussed it. Controlling of gas exchange in the cell, controlling of nutrient and toxin exchange, contain internal environment versus the external. That microtubule system is evaluating the electromagnetic energy. It's evaluating the pH. It's evaluating different ions. It's interacting with the environment, and it's processing that information and emitting the different frequencies it needs to do to remain in homeostasis. So the final cell function, reproduction. This one gets pretty interesting. In conventional biology, this whole centriole-centrosome structure, the only time you really learn about it in biology is about division of the cell. So these coordinate the separation of the DNA and the splitting of the cell, organizing all the different organelles within the cell, reproducing them into two functioning cells at the end. Very big topic. There was a researcher, I'm going to say his name was Dershowitz. I'm not sure how you pronounce it, but I call him the Onion Man, his famous onion experiment. He basically was working on how do cells know to divide. You got this cell sitting there. At what point does it decide, well, it's time to make two of me? The DNA? Again, when you get into conventional science, they don't have answers for any of these things. There's chemicals, some chemicals. What chemicals? They identify certain chemicals they believe are responsible for it, but if they put those chemicals in with a cellular culture, they can't get them to reproduce purely by chemicals. That's what Dr. Dershowitz was working on. He was trying to figure out what is it, what chemical, what makes a cell divide. He couldn't figure it out using chemical stimulants. What he then thought was maybe there's something unseen that's going on in cells. This was back in the early 1900s, by the way, he was doing his research. Maybe there's an unseen force. Maybe there's something about the cells where there's a way it communicates that isn't chemical, isn't something we have any type of comprehension of at our level of science. So he devised an experiment called the onion experiment. So an onion has roots. Those roots are dividing very rapidly. There's lots of mitosis and growth in the roots. He was wondering if there was some type of an invisible force that traveled down that root. So in order to test his experiment, he took another root and he put it perpendicular with the theory of this root is going to induce mitosis at the mid-root of the growing onion. If this is true, he should see that the cells that are facing these onion rootlets are going to have mitosis at a higher rate than the cells on the opposite side. Guess what he discovered? It worked. He found that this root started to actually curl because one side of the root was growing faster than the other because it was getting induced into mitosis from the other root. The guy was kind of genius. He decided, well, what is this? What is this unseen force? So the first thing he did was put a lead blocker in between. He found that when the lead blocker was in between, this no longer happened. That induction signal was not reaching this root, not increasing the mitotic rate of that root on that side. Then he started using, and then, by the way, that blocker is going to block all electromagnetic energy. Then he started using different crystals, different glasses, different ways to block certain frequencies of electromagnetic energy. He concluded that what was inducing that root to grow was UV light. Pretty profound if you think about it. That pretty much went nowhere, did it? Science never took any of this research, and there's plenty of research on biophotonics that occurred after this, especially in Europe, where that Rockefeller system wasn't taking control of the system at that point and knocking all that grant money away from anything that wasn't biochemistry and petrochemical based. So other countries have done tons of research on this topic where they're finding all these different frequencies. So it's kind of amazing because I told you earlier, this guy is the antenna system. So there must be something coming indirectly from the system, from the quantum system, from a central higher area that's controlling how the organism is functioning as one unit, that is telling these guys, we're going to divide. You need to separate. We need to move to this side, and we need to move to this side, and we need to produce a whole new centriole system. And that's exactly what occurs. So once those guys separate out, they start organizing their microtubules, they split off the DNA, and they start organizing the cell and dividing it up 50-50. So pretty interesting because you got two topics here. You got the mitochondria dependent on electromagnetic energy. You've got cell division, most probably related to UV frequencies inducing it. So quite exciting. My book is much more detailed. My other video series is much more detailed, but I'm hoping that I gave you a basic understanding of it. And like I said, after this little conversation video I had with you guys today, you know more than most doctors do about this topic. Most doctors all think that this is all random. Everything that's happening within a cell is just chemical reactions governed by enzymes that work in a magical way. That DNA unwinds itself and produces certain proteins just in a magical way. That cell knows it's going to be a liver cell, so it unwinds that segment and produces the proteins a liver would produce. No answers in this. As a student, when I was learning it, I just kind of had it in the back of my mind. I'm like, well, there's a higher power. Somehow God's controlling all this, which could be is the truth. But God is controlling any of this through electromagnetic energy. The system is interfacing with the unseen and the seen through electromagnetic energy. So I hope you enjoyed this. I look forward to our next video on oxidative stress. We're really going to hammer down and you're going to understand that mitochondria better than most doctors do after we're done. So thank you for your time.